As we have seen, DNA forms a DNA duplex with two complementary strands. Watson and Crick had already suggested that this might explain how replication works, namely by making two identical copies of the old strand. Soon after that, in an experiment performed by Frank Stahl and Matthew Messelson, showed that this is indeed the case. It is occurred as shown in this diagram, where you have two potential black strands which are then copied to make one black and one orange strand each. Messelson and Stahl showed that each new DNA contains one parental and one daughter strand, black and orange respectively, hence the term semiconservative replication.
So does that mean that each daughter cell contains one black and one orange strand?
That is indeed the case. And this experiment ruled out other models, for example, that a daughter cell would have two black and the other one two orange strands. Replication is a complicated and highly regulated process to make sure that DNA is copied accurately and that each nucleotide is copied only once per cell cycle.
Do you think it's too complicated to understand?
I do not think so. I think you could understand it, but for today, we will only look at the basic steps here. The first enzyme in this process is called a DNA helicase, and it separates the two strands like this. This gives rise to two single strands that then the polymerase can use to make new DNA.
So what is the reaction catalyzed by DNA polymerase?
The DNA polymerase extends only one direction, 5' to 3'. It goes so on both strands. And it extends the primer from a 3' hydroxyl group. It uses nucleoside triphosphate as a fuel, which it adds to an elongating primer. The primer is essential for this reaction, and it is made in the cell by a special enzyme called primase. If you want to make a primer in the test tube or use a polymerase in the test tube, we can use a synthetic primer.
We will see this example in the polymerase chain reaction in the next unit. We will also tell you how we make a DNA molecule chemically to use as a primer.
The fuel for the building blocks, as I told you, is the NTPs. These are the ATP, the CTP, the GTP, and TTP, which pair with the complementary base to extend the primer. In the active site of the polymerase, we will have the following components. We'll have the template, we'll have the primer, and we'll have the nucleoside triphosphate. This triphosphate here is the leaving group, and hydrolysis of one of this high energy phosphorus-oxygen bond is one of the driving force of the reaction. We can illustrate this with the following flow of electrons from the hydroxyl group here, and then as a leaving group, this pyrophosphate. This process is continued many times to complete the process. Now let's have a look at the polymerase structure. We have here, often representing this as the right hand. We have the fingers in blue, the palm in red, and the thumb. So we can represent this as a right hand. And you can see here, there's a cavity in the middle.
So is this where DNA goes?
It is indeed. You can show in the next slide, I can show it to you here in black and purple. The fingers open and close with each addition, and this helps to move the DNA along the active side of the polymerase for the elongation. DNA polymerase was discovered by Arthur Kornberg in the 1950s, shortly after Watson and Crick showed what DNA looks like. This discovery showed how an enzyme can carry out DNA synthesis and semiconservative replication. However, the importance of DNA polymerase is much greater than just for understanding how a cell works. DNA polymerase is perhaps the most important enzyme in biotechnology. It is needed for all forms of DNA sequencing, for example, for the Human Genome Project that has given rise to precision medicine that is so important today.
It is also needed for the polymerase chain reaction to amplify DNA. I will tell you about it in the next unit.
As such, it is a key to biotechnology. The importance of the discovery of DNA polymerase therefore far exceeds what Arthur Kornberg could have predicted in the 1950s.